All right, well, good evening. My name is Jessica Flack, and I'm on the faculty up on the Hill at the Santa Fe Institute. And it's my pleasure tonight to introduce Ian Cousin. Before I introduce Ian, a few orders of business. Tonight's lecture was made possible by generous support from the Aline and Jerome Lapidis Foundation in honor of the Santa Fe Animal Shelter. And for this support, we at the Santa Fe Institute are really most grateful. I'd also like to note that every year during these weeks of June, the Santa Fe Institute holds its Complex Systems Summer School, and we bring some of the best and the brightest graduate students and postdocs from around the world to Santa Fe to gain exposure to the concepts and tools of complex system science. And as you might expect, this is one of our flagship programs, and I'd like to welcome the students that are here tonight in the audience. Okay, tonight's lecture is on collective organization. It's about how individual behavior and decision-making produces group dynamics, and how beautiful, orderly, three-dimensional structures, like schools of fish and flocks of birds, that would seem to require complicated communication and coordination emerge from relatively simple decision-making rules by individuals implementing strategies using typically local information. Now, often, this is viewed as a problem that's unique to animal behavior and specific to organisms that form collectives moving through space. However, when one steps back, one realizes that this problem of collective organization is perhaps one of the most fundamental problems in the history of life. Our bodies are collectives of cells, our brains are collectives of neurons. And sometimes the collective behavior is relatively easy to see, and sometimes, as in the case of social structures, it's an invisible reality that can feed down to affect individual decision-making and rules, and in the best of cases, facilitate individual behavior. So whether invisible or beautifully visible, relatively little is understood about how coordinated aggregations of components, whether these are cells, or fish, or monkeys, or humans, form. And Ian Cousin is working towards remedying this deficit. He runs a collective animal behavior laboratory at Princeton, where he's assistant professor in the Department of Ecology and Evolutionary Biology. He received his PhD from the University of Bath in England, and he was recently named by the magazine Popular Science as one of the 2010 Brilliant 10. And as Popular Science writes, young scientists who are dreaming up the solutions to some of the world's most vexing challenges, and I think that pretty much sums him up. Ian gives a great talk, I've heard him before, and it's my great pleasure to welcome him here tonight.
Thank you very much. It is a wonderful opportunity to come here and speak to you today about my research on collective behavior. And I've been fascinated by animal groups, organizations such as schooling fish and flocking birds for a long time. Let's just see if we can get this. I had this problem yesterday as well. Sorry, I'm not going to upgrade anything. If it doesn't work, it doesn't. Oh, here we go. So here we see silver-sided fish schooling on a coral reef, and shortly you'll see European starlings flocking. And this type of amazingly coordinated behavior is actually, in this case, it's among unrelated individuals. And so from an evolutionary perspective, that's quite challenging. Why are these unrelated individuals behaving in this way? It almost looks like they're a single mind. And indeed, in the 1940s and 1950s, this was how people thought about these types of groups. And what we're beginning to realize, and I stole this photo from Qantas In-Flight magazine, and I wish I'd remembered to take the photographer's name. I always feel guilty when I show it. But it's a wonderful picture, and I've added these yellow arrows here showing you a sort of vortex that forms. These are sheep crossing a bridge. And you can see that the individual sheep tend to align the direction of travel with near neighbors. You can also see that they tend to try to maintain a minimum distance among themselves, except when they're sort of crushed together as they form this moving group over the bridge. And here it's physically the shape of the bodies and frictional forces that causes this pattern to form. Now, these individuals, you get a sense when you look at this image that they're trying to move in this direction. So organisms such as cells or animals in groups, as I'll show you today, are actually often reconciling this local tendency to interact with their neighbors with some sort of goal-oriented behavior. And as I mentioned, this really does span scales in biology. From these are the cells that actually heal your wound. If you cut yourself, your cells will collectively migrate to help heal that wound quickly and stop infection setting in. So collective behavior is both, within us and around us. And biologists have studied to a great degree collective behavior in these organisms. These are ants. And here we're estimating where the ants are leaving a chemical trail. And this is how they communicate with each other. They lay the trail, others detect that trail, tend to follow it, and lay their own trail. And what happens at the level of the colony, I'm now going to zoom out, and here are the ants entering the arena. This is time-lapse footage. This is a real colony forming this wonderful dendritic foraging trails in this environment. There's no food here in this experiment. The ants are searching this environment. But these are related individuals. They're very closely related. So in terms of the social insects, you can see that the individuals are doing this for the benefit of the collective. But as I mentioned before, when we look at schooling fish and flocking birds, they are selfish individuals. Now, we too, I'm afraid to say, are selfish individuals. So we've also been doing experiments with human crides, which I'm not going to talk too much about today, but we've developed software that can track the positions, the motion, and also where you're moving your head. And there are all sorts of collective behaviors that go on within our own society, which is, I think, one of the reasons we're so fascinated in it. So today I want to talk about 4 main themes. I want to talk about information transfer in animal groups, and that relates very strongly to collective decision-making. Then I'm going to talk briefly about the evolution of migration, so how we can use these understandings of the mechanism of how groups form and how groups move to understand new evolutionary principles. And finally, I'm going to discuss some work we've been doing trying to understand why do locusts swarm. And so again, I'm very lucky to work with the BBC. So they've taken my simulations and put wonderful graphics on it. This is real footage. These are real fish. But shortly, this will go into a simulation now. And this is just showing you, when we're looking at this multitude of interactions, I'm a biologist. I want to get inside the head of this individual. And it's very difficult to do so. How do we understand how all these interactions scale to collective behavior? Well, one way that we can do that is using computational modeling. We can make virtual fish in the computer. So here are 2 virtual fish. They have positions, which I call C, and they have velocities, which I call V. And then we can represent the types of hypothetical interactions that we think go on. Now, why do we do this? Well, until recently, there was a dearth of available experimental data. And so I'll discuss that in more detail shortly. But when I started this in about 2002, there was literally no data. So one has to start somewhere, so create a computer model. So this equation here just represents the fact that when these organisms come close together, they tend to try to push themselves apart again. They don't want to collide with each other. Similarly, as you saw with those sheep, individuals tend to be attracted towards each other, which is what this part of the equation is stating, and they may or may not tend to align their direction of travel with their near neighbors. So we can characterize those types of rules and put them into a virtual model inside the computer and then run that to see what types of patterns and what types of behavior we get. And so this model that I'm going to show you is in 3 dimensions. So individuals move through 3-dimensional space represented by this arrow. And I'm representing their behaviors in these zones. So the ZOR is the zone of repulsion. So if individuals are in that zone, you want to move away from them. The ZOA is if you get isolated from the group, well, that's dangerous. So you want to be attracted to other individuals. And there may or may not be this tendency in the ZOO to align your direction of travel with near neighbors. Now, what I'm going to show you now is what happens when I play around with the ZOO, this tendency to align with neighbors. What if we assume there's no alignment? There's only local repulsion and a longer range attraction. So here we see just the local repulsion and longer range attraction. Well, we get a mosquito-like swarm. So this is a static image from a mobile simulation, and the individuals are moving around in this quasi-stable state, but the swarm doesn't really go anywhere. Now, what I'm going to do is I'm going to introduce a very local tendency to align with neighbors, a little bit like you saw with those sheep. And look what happens to the group structure. I introduce this zone, and we get something quite surprising. The group now forms a torus, a sort of bagel formation, where they perpetually rotate around this empty core. And what's important to note here is there was nothing in my equations of motion, in my equations of the behavior of these organisms, that told them to go in a circle. This is an emergent outcome from following these types of behaviors. And interestingly, if you either grow the size of the zone of orientation further, or you wait for a long time, the group can also exhibit another collective state, which is this dynamic, fluid-like behavior that we're very familiar with, that sort of amazes us when we look at bird flocks and fish schools. And these are my own graphics, not the BBC's, as you can probably tell. But this is the torus formation, this rotating state. And this was actually the first computer program I'd ever written. And so I assumed there must be a bug in the code. And so I spent weeks looking for errors in the code. I couldn't find any. And then going to the literature, I found lots of examples of these patterns in nature. So what's nice about this type of modeling is, one, I wouldn't have expected to have found these patterns. And the other thing is my model predicts there are only 3 fundamental patterns that the groups should exhibit if they are following these types of rules. So when I moved to Princeton about 3.5 years ago, I was really keen to move beyond modeling and actually develop an experimental system so I could try to interrogate the real world, the real dynamics of a fish school, to the same level of detail that we can within these types of models. And so I'm literally going to show you a video from day one. The fish just arrived. We put them in a tank that's 7 foot by 4 foot, and it's shallow water. And we're looking from above. And you can see these fish. This is sped up a little bit just to save time. But you can see them in the parallel group state. And shortly, you'll see them transition into the rotating state. So I was pretty astonished that this very simple model predicted the behaviors that we actually see in reality. But what we can now do over the last few years is we've developed software that can track hundreds of individuals, maybe even thousands of individuals simultaneously. So instead of just resorting to virtual fish, we can now get really good data of how the real fish move and interact with each other. So we can get the positions and the velocities of all of these individuals 30 times every second. And this is Haishan Wu, a Chinese graduate student that's developed this. And it's really quite remarkable because, look, when the fish overlap with each other, which is a notorious problem when trying to do this tracking, we can still keep track of them. This is just tracking one fish, but of course we can track all of the individuals within the group. So this gives us really good, detailed information about how the individuals behave. And what we've also been able to do, and this is very, very new, is we've been able to develop algorithms that can actually take those types of data, those movement data, and extract the interactions from the real system. So it's called solving the inverse problem. And you may think, well, that's kind of fun to do with fish. But the important point to note is we could also do this with cells or with ungulates. Or if you have the precision of data using GPS, for example, we could then now extract these interaction rules. And so we can track the fish, how they're interacting. Actually, I'm really happy. This paper was actually accepted just this afternoon for publication. So thank you. Well, we should really thank Yael Katz, who's the first author of this paper, a postdoc in my group, who really pioneered this work. And so what we can now do, this is not a simulation anymore. This is representing the position of a focal fish. And what these arrows represent are the forces, the social forces it feels from other fish. This is extracted from real data. The colors represent where the fish tend to like to space themselves with respect to each other. So what this shows is if an individual comes very close, the force this fish feels is a strong negative force, i.e. it will decelerate to avoid collisions. If something comes quite close behind, this is a strong positive force, it will accelerate. Similarly, if it gets left behind, that strong positive force means it accelerates. If it leaves the other individual way behind, what it will do is it will decelerate. So we can look at these types of interactions for the first time. And I have wonderful graduate students. Colin Twomey is a computer scientist in my group, and he's extended this software. So not only can we track the movement of the fish, but we can track where their eyes are and then reconstruct their visual fields to see what the fish are seeing. So I'll zoom in on this. So you can see where the estimated eye positions are. Again, 30 times every second, we can then really work out what these fish are seeing in their environment. So we can really build up a social network, a topology of interactions that's not just based on proximity, but is based on the real information that the fish are using to guide and coordinate their behavior. We're also developing this technology in 3 dimensions to use with primates and also with large birds, so we can actually see where organisms are looking in 3-dimensional space. And when we put 1,000 fish in our tank because we had to clean all the other tanks. So what do? We put all the fish in this big tank, and then, look at what they did. It's just astonishing. They always surprise me. And you can see these amazing waves. So many times these waves don't propagate. The fish will startle, but it will just dissipate. So you get these little avalanches. But every now and again you get this big avalanche that sweeps across the group. And so you really have this wonderful dynamical system in the laboratory that we can get all of this information from. But we also want to manipulate it. We also want to play around with it to see how information flows in the group. Because one of the remarkable things that you see within these fish schools is this incredible synchronous response. And as I mentioned to you before, in the 1940s and 50s, people thought they must be telepathic. They must be able to read each other's minds to coordinate their behaviors in this way. So this is the same simulation I showed you at the beginning, but I've added a predator and one more rule, which is quite a sensible one: run away from the predator if it comes close, or swim away, or fly away. And you can see now, I'm going to pause it and rotate around, and you can see the sort of vacuole around the predator. But the fish here are also moving away from the predator. They don't even detect it, but because they're responding to the others that do detect it, you can get this wave of information propagating across the group. And these waves can actually, in oceanic fish, these very large pelagic fish schools, can travel literally kilometers. So it's a wonderful information processing system, and I love showing these penguins. Look at them, they're just nuts, right? So beautiful. So we don't have penguins, but we have a robot predator that we've developed. So this is being controlled by magnetism. So we have an autonomous robot underneath the tank, which is controlled through Bluetooth. And we can actually, I'll show you shortly, we can track these fish, and the predator can make its own decisions when and where to attack. So we can begin to ask questions, both about how the individuals behave. Oh, maybe this isn't going to... Let's see. Well. So what we have here is we can track the motion of the fish, and this can actually feed back into the motion of the predator. And so you can see these fish, these 2 schools are isolated. They're pretty scared, actually, of this predator, so they form a very tight group. And this is early days. The predator is not very good at attacking at the moment. We're working on this. But this is just to give you an impression of what a predator can do. So it's making its decision. It's taking in this information. We're reconstructing its visual field, and it tries to attack the group. We can also get these types of data from the field, in the real world. This is a real estuarine system. It looks like you're looking from the top down to the bottom of the ocean or something like that, but you're not. You're actually looking from above. So this is all 1 meter depth, all the way across. And we're seeing these predator fish attacking these schooling fish. So we can really observe these types of information transfer, and then using our tracking software, we can track the motion of the predators and the prey to really understand what's going on in a natural environment. And these estuarine environments are really getting damaged by human influence, and so we want to have this long-term survey of how these fish behave. And really what I've been hinting at is, can we think of these groups as forming some sort of collective mind? Can the group have an awareness of its environment that is, more than the awareness that any individual has? Can the group make decisions based on information that no individual knows everything, but somehow the group can come to an accurate consensus? And so to start off, I want to talk about leadership within animal groups. So when we deal with, again, I'm going to show you pictures of fish, but please think about this more generally. These could be cells or birds or other animals. So in this case, this individual here, it knows what's going on. It knows there's a food source over here, for example, or it sees a predator down here that the others don't see and it wants to move away. So we're not telling you exactly what information we're encoding in the model. It's a very general framework. Okay, but this individual has some information and it wants to go this way. Now, these individuals don't have any information. So how does that individual propagate that? Does it need to signal that I've got information, follow me? Do others need to recognize who has information and who does not? And also, if these are unrelated individuals, why would you actually share this information with others anyway? So again, we can use computer models to give us a good understanding of the types of principles here in terms of leadership. So this is an even simpler model than the one I showed you before. We're now just in 2 dimensions, and we now just have 2 zones. And the reason is we always try to find the simplest possible explanation for complex phenomena. Okay, the model could be much more complex and much more biologically realistic and give similar results, but we wouldn't fully understand it. So that's why we use these simple models. And so this individual here, it knows that it wants to move in this direction. Now, this is an important point that I've colored this individual bright red. Now, please remember from now on that when I show you these colors, this is so you can see and I can see what's going on. Like who has information and who does not, and what type of information they have. But in the model, nobody knows the information that others have. They start at random positions, random orientations. They cannot tell others what they're thinking or what they're doing. So it's the minimum set of assumptions, okay? So the color's just for us. And so this individual has to reconcile its social tendency, its desire to school with the others, with its desire to want to go and do something else, to go and find a food source, for example. And it reconciles this with a term that it's going to come up again and again and again. And it's very simple to understand this. You don't need to think about this equation. But this term is called omega. That sort of funny W is an omega. And that's the strength to which it believes, or it has that strength of opinion. So if the omega is very low, it's not very hungry. It doesn't really need to go anywhere else. And so it will largely school. If the omega is very high, of course, it has a very strong opinion and really wants to go in that direction. And of course, it may leave the group behind if it's particularly strong. And so the first question we wanted to ask ourselves, well, can individuals, without signaling and without individual recognition, transfer information within these groups? So here's one individual, colored white, trying to guide the group along the x-axis there, but it's not able to do so. But if I put in 5 informed individuals, the big green cross just shows you the center of the group. If I put in 5, you can see after an initial period of disorder, the group kind of goes in the right direction. You get a sense that they're transmitting some form of information, but perhaps not most accurately. But if I put 10 informed individuals in my group of 100, suddenly and spontaneously, the group will go very accurately in the correct direction. Again, no signaling, no individual recognition. So we can quantify this accuracy as a value from 0, which is completely random motion, to 1 being absolutely perfect motion, and they're never perfect because there's noise and there's error in individuals' judgments. But you can see let's look at a small group now, a group of 10. As we increase the number of informed individuals, the proportion of informed individuals, the accuracy of the group motion increases asymptotically. Now this happens for bigger groups also. And if we look at the largest group here, so for the small group, for it to be around, say, 85% accurate, well, we need around half of them to have information. But if we look at the largest group, a group of 200 in this instance, for the same level of accuracy, around 85%, we need only around 5% of the individuals to have information. So as groups get larger, the proportion of informed individuals you need to transmit information to that group gets much, much smaller. And we can scale this up, I'll show you later, for migratory groups. But what about differences in experience and motivation? So far, all of these leaders have wanted to go in the same direction, but we're all too familiar with individuals not agreeing with each other. And similarly, within animal groups, there are often disagreements about what to do. So can the group come to a collective consensus? So in this case, some individuals want to go one way, others want to go another way, and others haven't a clue what's going on at all. And so what I'm going to do is I'm going to keep my group, one of my groups, to want to go along that x-axis. Then I'm going to create another group that disagrees with them. Now, they may disagree by a very small amount, only say 20 degrees. Or they may disagree by around 90 degrees. Or they may disagree by 180 degrees, where they actually want to go in opposite directions to each other. And I can then run thousands of simulations for all of those degrees of difference in opinion and ask where the whole group goes. And the white line just shows you the average decision between those two subsets. And so I'm going to set it up with 100 individuals, 5 want to go 0 degrees, and 5 of them disagree with them. And I'll run this, and I find out where the group goes, and that's shown by this color plot. So I'll take you through this. So what we can see is below a critical difference of opinion of around 135 degrees, the whole group tends to go the average direction. They split the difference. But above that, something interesting happens. We get what we call a bifurcation, a sudden transition in collective behavior, whereby half the time, this is not the group splitting, it may look like the group splitting, but half of the time the group chooses this direction, and this 5 wins, and half the time the group chooses this direction, and that 5 wins. So the group is making a consensus to go one way or the other. Now, if I put in just one more extra individual into one of those groups, so 1% of total group size, so we now have 5 want to go one way, and 6 disagree with them and want to go the other way, what happens? Well, without being able to talk to each other, without being able to do mathematics, the group spontaneously and accurately reaches consensus. So individuals have local, noisy information. They can't do anything like counting like we would do to make these types of decisions, and yet very suddenly and spontaneously they can do this. And so they can solve problems. Effectively, they can perform computations just through the kinematics of their motion, just by moving and interacting in this way. And I won't go through all of the studies, but a whole bunch of studies have been performed since this paper came out in pigeons, and in humans, and in fish that support this view. And I'll discuss some of the work in humans, because I originally wanted to test this idea with fish. But it turns out that fish are really hard to work with. And it was much trickier than I'd anticipated. And so we turned to a much simpler organism, which were undergraduate students. And so we could put our undergraduate students in a ring here, and we point them at random directions. Okay, so that represents our random starting conditions. Our groups are relatively small because we can only use each student once, and we need to get enough replicates. And the students have cards, and the card either says you have to evacuate this area. There are these exits that are much further away. You have to evacuate this area, stay with the others. And other people have cards that say you have to evacuate this area and stay with others. The safe exit is exit 13, say, or exit 12. Okay? But they're not allowed to talk to each other. If you're in an evacuation, you don't necessarily stop and go, "Excuse me, do you know which way to go?" You tend to maybe just follow where you see others going. And indeed, this very simple rule means that we only need one student to have correct information for the group to be very accurate at escaping through the correct exit. And it's even more accurate with 2. We can then set up conflicts. So there are 3 students that think one exit's safe, 2 that think another exit's safe, and some that don't know what's going on. And we can see that, again, the majority dictates group motion, even though there's no verbal communication. German television were very interested in this and wanted us to find how many or what proportion of Germans do you need to guide 200 Germans out of a very large gymnasium. I'm not quite sure why. But anyway, so we performed these experiments, and we put hats on the individuals, and we knew which hats were associated with different types of information. It was kind of interesting because it was exactly what the model predicted, absolutely spot on in terms of the proportion that we needed. But we can do a bit more serious things with this. We can begin to develop software that can track the motion of pedestrians in crowds, as I showed you at the beginning, and also simulation models that can simulate how people move and how people behave. But to come back to this model, now some of you may have noticed that 135 degrees was the point at which the group started making these decisions. But up to that point, they chose the average. Now, if I want to go this way, and you want to go this way, and we all end up going this way, then it's bad for both of us. Okay, but in actual fact, when you deal with the real geometry, moving through real space, when the group is far away from the 2 targets, so the white want to get to the white target, the red to the red target, when they're far away, this angle is going to be small. The further away they are, the smaller it is. But they're always going to reach the point of the 135 degrees, and therefore be able to make a consensus decision. So here there's an equal number of white and red individuals, and the green ones are the ones that don't know what's going on, and in this case, they choose the red target. Now I add just one more individual, one white individual to the group, and I hope you almost see from these visualizations how local and noisy the interactions are, and yet over 99% of the time the group can accurately determine what that subtle majority is. But what if you're really unwilling to give up on your opinion? If your omega is really big, well, then the group can't come to consensus, and they will tend to split apart. And of course, this is common in nature, and it's an interesting region to be in. But interestingly, also, for intermediate levels of omega, the group exhibits this type of trap-lining behavior, where they sort of check out the different options before they make a decision. And we actually see this also in reality. And so I want to talk to you about something very new. I've just submitted this paper about democracy in animal groups. Well, you may think about democracy more commonly within human societies. Across the world, it's something that's convergently occurred, is people tend to make democratic decisions, whether it's in committees or governmental panels or whatever. It's very, very common. And another thing you may know about democracy is our democracy is an adversarial democracy. Right? We don't necessarily agree with each other. You know, not everyone is the Democrat, not everyone is a Republican, yet you choose democratically one government. And similarly, within animal groups, there can often be great costs to not coming to consensus, to these groups splitting apart. And so I'm going to specifically discuss the case of consensus decision-making and democracy in groups today. So as I mentioned before, there are often conflicting interests among group members, and yet failing to achieve a consensus, failing to actually decide on a government, or to decide on an action plan in a committee, can be very costly. Individuals may be susceptible to manipulation by opinionated or extremist individuals. And previously it's been argued in the social sciences and also in biology that those who are uninformed or have weak preferences are particularly vulnerable to extremism, are particularly vulnerable to being influenced by the opinions of others. And so in human society, what have we done to inhibit extremism? Well, one obvious example is voting. Voting promotes fair representation because it removes the strength of your opinion and it turns it into a single value, a single vote. So one person, one vote. So it doesn't matter how strongly you believe in it, you still get one vote. Okay? And so this results in decisions being based on the number of votes cast as opposed to the strength of voter opinion. However, in many animal groups, there's a limited means of conveying a vote, as you can imagine. There's no global overview, there's no counting, as I showed you before. They don't need to be able to count. But there is no global overview of votes, and only relatively local interactions. So does this concept of fair representation make any sense in biological systems? So I'm now going to show you the same type of model, those individuals moving towards two different targets, and omega-1 represents the strength of the majority, and omega-2 represents the strength of the minority. Now, there's no minority here, there's 1 and 1, but I'll come to that shortly. So here we have a majority, right? There's 3 want to go one way, and there's 2 that want to go the other way. So everybody has information, but they disagree with each other. Now, what should happen? Well, what I just showed you before is that the majority should be able to win out, and the group come to a consensus to move in that direction. But what if the minority just increases the strength of their omega? Increases the strength of their opinion. They really want to go that way. They really want to influence the group and go that way. Well, what happens now? Well, what we find is as we increase the strength of minority opinion, initially, if they have the same strength of opinion as the majority will win. 1 means the majority wins all the time. But as the minority increase the strength of their opinion, they begin to control the group. Now here it's 50-50. Half the time the majority wins, half the time the minority wins. But if the minority become really strongly opinionated, they could dictate the group direction. So it seems to work that if you increase the strength of your opinion, you can influence the group more strongly. But this is with no uninformed individuals. Everybody has information in the group. What about if I put in stupid individuals, or individuals that don't know what's going on or don't have an opinion? Well, something really interesting happens. So here the minority has a stronger opinion. Not so strong that they win all the time. The majority is still winning 60% of the time, but the minority wins 40%. But if I add my naive individuals that don't know what's going on, they return control strongly to the majority. And then eventually they'll saturate. So I can increase the strength of my minority so they become even more opinionated. Now they're winning, the minority are winning almost 90% of the time. But add stupid individuals, and it flips control back to the majority. And I can take this all the way down to now the minority is winning almost 100% of the time. And again, add individuals that don't know what's going on, they don't even know a decision is being made, and they flip control spontaneously to the majority. So effectively, they're promoting a democratic consensus, and they're removing the strength of opinions from this. Okay, and so in this simulation model, I can show you for different types of majorities and minorities that this result holds. But one of the problems with simulations is we can't really understand fully whether this is a generic result that relates broadly to different types of systems. And so what we've done is we, inspired by this model, we then developed a more formal mathematical tool set to try to prove this result. Okay, so we want to see, is this a general principle? So we decided, well, we're thinking about opinions, we're thinking about voting, I've been alluding to that. Why don't we actually use a model that specifically simulates how we ourselves have opinions and how we influence each other's opinions? And I won't go through the mathematics of this. I'll just describe to you very simply what we did. So, individuals have opinions on one of two mutually exclusive options. Okay, so just for simplicity, let's say you're Republican or you're Democrat, and there's just two exclusive options. And we're going to, as we did before, have a strongly opinionated minority that really believes in their viewpoint. A less strongly opinionated majority. You can almost think of this as an extremist group. And individuals that begin with a weak or no opinion at all. And so what we can do is simulate how, when you're talking to other individuals, they influence your beliefs. And similarly, as you talk to them, you influence their beliefs. So there's a feedback. And if you have a certain belief and everyone you meet disagrees with you, that can then flip your belief. Okay, so I won't go through the details, but what we found was with very, very broad range of parameter space, with very few assumptions, we found exactly the result that we saw in the simulation. If the minority is controlling the group behavior, we can add naive individuals and it will flip control back to the majority. The nice thing about this model is it's analytically tractable. So as I mentioned before, we can actually prove this result. And you don't need to worry really what this means, but this basically shows that the only stable state without naive individuals is minority winning, and then suddenly above a critical threshold of uninformed individuals in the population, there are two solutions. The majority can win or the minority can win. And as you can see in the numerical simulations, the majority tends to take control. So this branch is the one that's used. So this is suggestive that this is a general principle. So uninformed individuals, by removing the influence of opinion strength, enforce fair representation. Our theoretical studies, therefore, both the simulation of the swarms of the flocks and our simulation of how people interact and influence each other's opinions, make a single prediction, a strong prediction, which is that uninformed individuals should inhibit the influence of a strongly opinionated minority and return control to the majority. Now, I love playing around with these simulation models, but I'm a biologist. I never quite believe it unless I can do a real experiment with real organisms. And by this point, I was getting a little bit better with my fish, so I decided to try again with a fish. Okay, and so because of, again, we need a large number of replicates, we can only use each fish once and so on, within the laboratory, I decided that I was going to look at 0, 5, and 10 naive individuals because the simulation predicted that this should be a very steep response. So I should be able to really strongly influence the group dynamics. And so this is showing one of our experiments. So here are the fish that are being tested. Some of them want to go to this blue target. They've been trained. Fish are very clever. They've got very good memories. They can maintain this information for months or years. And some prefer this yellow target. And you can see, we can just then let the fish go and see what they do. So we can read it out. So this is a little bit like, it's a real simulation. It's the real organisms. And so Christos Wanu, my postdoc, did these experiments. And so 6 want to go one way, 5 want to go the other way. The target the minority want to go to is inherently preferred by the fish. Many organisms, including humans, have intrinsic biases, and we exploited that to create this strongly opinionated minority. The uninformed individuals don't have any bias or even knowledge of the targets. As I said, we're going to look at 0, 5, and 10 naive, and we did a whole bunch of experiments that took about 3 months. And so this is the prediction, the general prediction. And as you can see, we set it up such that the minority, when there's no uninformed individuals, the minority will tend to win out. They have a stronger opinion. But we can then add in 5 individuals that don't know what's going on, and we begin to return control to the majority. Add 10 individuals in the group that don't know what's going on, and we return it to the majority. So just as we predicted, this counterintuitive prediction that adding in naive individuals returns control to the majority really works within this animal group. And so uninformed individuals inhibit extremism and promote democratic consensus decision-making. So I would like to also talk about so I mentioned at the beginning I was going to talk about evolution. And so another thing we've been interested in is individuals, so far I've been training individuals to go to targets or to have certain opinions. But you can also get your omega on the fly from the environment as you're moving through it. So consider migrating animals that perhaps can pick up the magnetic field of the Earth. This gives them a cue, a rough cue as to what direction they should go in. Okay, so that's their omega. So instead of it being just fixed, you're now getting it from the environment as you move through the environment. And this could be visual information, olfactory. The model's very generic. And one simple way of thinking about it is in cells, because this is one of the best studied examples. Cells can detect chemicals, and the pathways by which they process that information to detect chemicals to climb gradients is well known. So when your body forms, for example, your cells are climbing chemical gradients. Dictyostelium cells, a sort of famous biological example, follow gradients. And you can also quantify to some degree the cost, because you have to run ATP. This costs energy to run. And so what we're going to assume is individuals are trying to explore, a range of different complex environments. And so, these environments could be these kind of plumes of chemicals in the ocean, for example. Or they could represent magnetic fields. So it's a very generic model. So now our individuals have the social tendency, I showed you before, but it's an evolvable tendency now. So I'm not telling them to group together. They can evolve to group together. They can also evolve to trust the environmental information that they're obtaining. So they can evolve sociality, and they can evolve gradient detection. And these are continuous traits. So you can evolve a strength of sociality or a strength of gradient detection capabilities. Well, how do the individuals behave? Well, as you can imagine, as you invest in gradient detection ability, as you invest in having machinery to detect that gradient, you get better at detecting the gradient. No surprise there. As I showed you before, this comes at a cost. So the more you invest, the more costly it is. And so what's the optimal strategy? The evolutionary stable strategy for an individual, well, it's pretty trivial. It's the benefits minus the costs. And so it's a single point here. So you think, well, fine. So we know how much you should invest in gradient detection ability. But we're forgetting something. We're forgetting about the social component. If you're a migrating wildebeest, do you really detect the Earth's magnetic field if it's costly, or do you just follow what the others are doing? So as soon as there's social information, you can exploit that. And so what's kind of interesting is, in actual fact, because individuals are detecting the information about the environment, like the magnetic field or gradients, and it's noisy, and no individual is absolutely perfect at doing this, the best thing they could do is to group together. And this individual thinks it's this way, this individual thinks it's this way, this one thinks it's this way. But if they group together, they average that all out, and therefore everybody benefits. That's the best thing for the group to do. But it's not evolutionarily stable. What actually happens over evolutionary time, so these are generations, and we're evolving these populations, so we're evolving 16,000 individuals using GPUs to do this. And you can see initially, yes, they evolved to start detecting the gradient, and they evolved to start detecting each other. Win-win, wonderful, what a beautiful scenario. Everyone's helping each other out. But you can suddenly see this catastrophic collapse here, where some individuals, they don't know what's going on, but evolutionarily they pick up on the fact that there's cheaper social information. So all of these guys or girls who are using the magnetic field, well, I can just turn that off and follow what they're doing. So I can exploit them. But of course, as everyone tries to exploit each other, then no one migrates. And so it becomes viable to then pay more costs to actually find the gradient itself. And so what we find, what's called the evolutionary stable strategy, are actually two strategies that have exactly the same fitness, exactly the same cost-to-benefit ratio. So we have a small proportion of individuals that invest in detecting the gradient, and a large proportion that actually don't bother. They just use the social information. And so as a biologist, you wouldn't necessarily notice this, because it looks like everyone is using the environmental information. But when you look at the model, they're actually moving this way. This is a static picture. You can see the leaders at the front are the ones detecting the magnetic field or detecting the gradient, and these are the others using social cues. And we can show in the model that groups can get very large and yet have a very small proportion of these leaders. And we think that this is important in explaining something you frequently see within cells, which are these leaders guiding these groups out. If you remove these leader cells, the group sort of collapses. You put them back and it starts migrating again. In tumors, These cells here are secreting protoplanin, which causes calcium oscillations. But they've got a job to do. They've got to push through tissue. They're paying a cost. And so we're beginning to understand or develop new understanding for the evolution of spreading of these migratory tumors. So what about context dependence? These are kind of silly individuals. What if they're capable of modifying the rules? Such as if you detect a quorum of individuals around you, maybe you turn it off so you avoid being exploited. So what if we have dynamical rules? Well, even with no cost to switching, we still see that the evolutionary stable strategy is for some individuals to detect the gradient and others just to detect each other. And so, unfortunately, this is sort of new research. So it makes a prediction about how migratory groups should behave. And unfortunately, we don't have data yet. But if you look at, I found a thesis by a student who interviewed elders in Dawson about caribou migration. And there's a sort of a cultural information within the community. They've been hunting these caribou for hundreds of years. And the elders pass on sort of laws to younger generations about how to hunt the caribou. And they always say, don't take the leaders. In fact, there is a law, let the leaders pass, which is based entirely on this cultural information. So, admittedly, this isn't any sort of scientific proof, but it's because no one's looked at this. But it's kind of intriguing that they're saying, if you shoot the leader, the rest get confused. And so, so you can really get these large groups. So it's suggestive, perhaps, that what we're finding within our evolutionary simulations of leaders and followers maybe actually matters in real migration. And what we can also do is, humans are really messing up many animal migrations. And so we can simulate over evolutionary time what happens if we increase fragmentation of our habitat. How are we going to affect migrations? Well, what we find is migrations begin to collapse. And if a migration is completely lost here, it doesn't matter if you bring the habitat back to a case where you still had migrants. You're not going to get the migration back. In fact, you have to bring the habitat back to such a rejuvenated state before the migration comes back that to all intents and purposes, in practical terms, it's going to be impossible. So we predict you have to be very careful when managing migrations. So changes in migratory patterns have been recorded in many natural populations. And in fact, I had to change my slide today because there was going to be a road that bisected the Tanzania, which we thought and others thought was going to affect the migratory path. Well, that plan, fortunately, has been axed as of this week. And so our model predicts, it's good news. Our model predicts a potentially bleak future for migrants. We have to be very cautious about how we handle these. Okay, I'm going to talk about another migrating species. And this one you may have less sympathy for. These are locusts. And I want to ask, when, where, and why do locusts exhibit large-scale motion, or insects exhibit large-scale collective motion? And what are the biological principles? Why do locusts swarm? And so I was inspired by these types of footage that you see on television. These dramatic swarms. But in actual fact, this is like a wildfire that's gone out of, I shouldn't really mention wildfires around here, but it really is a bit like a wildfire that's gone out of control. It's incredibly expensive. It's incredibly difficult to control them. And what many people don't know is locusts actually form swarms before they even grow wings. They only grow wings at the last stage of their life. So for several months, they hatch out. So this is time-lapse footage from our lab in Oxford. And they hatch out, and they dry themselves under the heat lamps in our lab, which represents the sun, and they start marching, wingless nymphs. And that's the critical phase. Well, why is this important? This one species of locust, the desert locust, I do my field research in Mauritania. I'll show you some pictures shortly. This one species of locust invades up to 1/5 of the Earth's land surface during plague years. So over multiple years, they can invade up to 1/5 of the Earth's land surface. In fact, there's a real danger in Yemen at the moment because of the instability there that locusts are going to go into plague mode. It's estimated by the FAO that they influence the livelihood of 1/10 of the world's population. It's an astonishing impact, and yet there's hardly anyone researching this. And in a review in Science, it was written, even after 50 years of experience, fighting locusts is more of an art than a science. And that's because the last time someone put them together in the lab to see how they swarmed was Peggy Ellis in 1954. And she published this in the Locust Bulletin, which of course no one reads. It's not on Google Scholar. She was a woman who was well ahead of her time. And so the real world expert on locusts is a guy called Steve Simpson at the University of Sydney. But he was at Oxford when I was there, and I worked with him. In fact, David Krakauer here also worked with Steve on locust problem at one point. And locusts, in fact, can't stand being near each other. Might sound surprising. They're actually shy, cryptic green grasshoppers that don't like to be near each other. It's only when you force them to come together that they change the way they look, and they change their behavior, and they form these swarms. And what Steve discovered was that the sight and smell of other locusts, or the contact with other locusts, physically the contact to the back leg, causes individual locusts to go from shy, cryptic green to this swarming black insect. So it's amazing. In fact, it used to be thought that they were separate species, but they're not. So it's a sort of two organisms in one genome with an ability to flip. So we wanted to investigate the gregarious form. I'll come back to solitarius at the end. So the form that forms these swarms. And so, as you'll see later on, it's hard to study them in the field, and these are not the smartest animals out there. So we created an artificial annulus arena, and the locusts will run around here indefinitely, thinking that they're in a desert environment. And so I developed software, which is a bit more primitive than the fish software, but it still was good enough for us to track the motion of these locusts and ask what's going on. And also the nice thing is it could track them for 8 hours a day and not complain about it. And so what we could actually do is reduce all of those data down to a single value. So 1 is the locusts going clockwise, -1 is the locusts going counterclockwise in the arena. 0 is they're just going all over the place. There's no order. And so if you look at low density of insects, well, it's a mess, right? And it's because they're like particles in a gas. They're bumping into each other, but there's no real order to the system. But as we increase the density, here you can see them going counterclockwise, but then suddenly they switch to go clockwise, and suddenly they go counterclockwise. The group has a dynamic now. It's like a fluid that can suddenly change direction. And above that density, in this case, the group went clockwise for the whole 8 hours. So we can see that changing the density has a dramatic effect on the swarm behavior. And it may seem surprising to you, but this reminded us of how magnets work. So magnetic particles tend to want to align with each other. And that's what gives a magnet magnetism. But if you heat it up, if you add noise, and these particles start jittering around with the heat, eventually you get to a critical point called the Curie point, at which the magnet suddenly loses magnetism. And so we took a model of magnets and added an extra thing that magnets don't have, which is self-propulsion. So I won't go through this model. It's just simple alignment with neighbors, with some noise, but you can move. And we were really astonished that, just fitting 2 parameters, we could get a very good quantitative fit between how this sort of fluid magnet behaves and how the real locusts behave. And we could also see the total time spent in the ordered phase as a function of density, the total number of direction changes as a function of density, really matches extremely well this very simple model prediction. But I've told you that the locusts align and how this local alignment can create sort of moving magnetic type swarms. But I haven't told you why they align with each other. And this was one of those nice examples in science where we discovered this purely by fluke. So I had Jerome Buell was the first author of that paper. He was my postdoc. And he was a Frenchman who always wore black. And he always stood very straight. And he didn't like mornings. And so I don't like mornings much myself, to be honest, but I would set the locusts up in the morning, and then 8 hours later, he would dismantle the setup. And so that was the way we did things. And Jerome would come in and stare at me and go, "Ian, you didn't count the locusts." I was like, "What?" He said, "You meant to put in 50, you put in 49. I was like, "Oh, no, sorry, Jerome." And this happened time and time again, and I was getting kind of scared of this passive-aggressive Frenchman. And I made really sure that I was counting correctly. And they were still disappearing. And with a colleague of mine, Matthew Collett, we decided that we would go through the videos and look to see what's going on. And what we discovered was quite surprising. The locusts would be biting and eating each other. So these apparently vegetarian insects were really aggressive to each other. They were nasty to each other. They'd frequently eat bits of the abdomen. And if you're a desert insect, this is disastrous. Sometimes they'd eat part of the body. Sometimes they'd eat the whole thing. So Sepide Bazazi was an undergraduate student at the time. Steve and I decided, well, we'll try and create little incisions here to cut the nerve. And we tried for 2 weeks, and we failed and failed and failed. And Sepide just decided, I'm going to do this. So she sat in this hot, sweaty lab that represents a desert environment and eventually found a way to find the nerve to cut so the locusts can no longer feel others biting them. And of course, she did control experiments where she located the nerve but didn't cut it. Well, if you'd have your abdomen denervated, it might really annoy you But fortunately, locusts are quite robust, simple organisms. And so with our tracking software, we could show that the way they moved, also the way they ate, the way they turned, was absolutely identical. They didn't seem to know that we'd cut this nerve. But when we put swarms together of individuals that had the nerve cut, they didn't swarm. In actual fact, if you compare swarms of individuals that have had the nerve cut with individuals that don't detect each other at all, so individuals in isolation, there was no statistical difference. But if you have individuals that can detect the biting behavior, they start marching, they start swarming. So here are individuals that can detect each other beginning to march, and individuals that can't feel the biting become quiescent and as if they're not detecting each other at all. Also, we know that they have a complex visual system, so we were surprised by this. So we did a really high-tech experiment where this is a normal locust, and Sepede has painted the front half of this locust's eye black with model paint. This one's got the back part of the eye painted black, and this one's completely blind, the poor thing. And this is affecting the flow of information. If the front part is blind, you can't see those ahead. If the back part is blind, you can't see those coming from behind. And what we found, if you look predominantly at the green and the orange are completely blind locusts. They can't swarm at all. But look at the green, very close. And that's the ones that can't see them coming from behind. Okay, there's also a slight pull from those ahead, okay, which we've actually estimated to be a fifth of the strength of this push from behind. But if you can't see others coming from behind, and you can't feel others coming from behind, you don't swarm, and you get eaten. So when locust density increases, the swarm may look cooperative. They're all marching together, but it's actually aggressive interactions that's the dominant factor. So they're effectively on a forced march. If you stop, you get eaten. So I decided to go to Mauritania to study these locusts, which, as you'll see, was probably one of the biggest mistakes I've ever made in my life. Some parts of Mauritania were beautiful, and we found locusts, which you think, well, if there's a plague going on, it'd be easy. But there's such a vast Saharan landscape that finding locusts is quite challenging. What's also challenging is, here's our camel, and these are the footprints. So we were tracking the swarms. They're 15 kilometers long, these swarms, tracking them by their footprints. That's me filming them. This is our camel I just liked. I mean, how often do you do research with a camel? But I'm not the best field biologist, as it happens. And so we ran out of food. And I'd actually been vegetarian for 10 years prior to this trip. And this is camel entrails hung in a tree, uncooked, drying in the tree to form a jerky. If you look closely, you can see flies laying eggs in it. And so we dried it in the tree and then ate it like jerky, and I became horrifically sick. And of course, there's no medical care in Mauritania, so I was just stuck there. And then what happened, as soon as I got better, this huge sandstorm came. So this is the Mauritanian colleague's tent. That's my $50 tent. This is the tree that we're drying our clothes in. We're very resourceful. That's also the tree that we hung the camel entrails in. And of course, we couldn't get anything but camel entrails because there was a plague going on. So it was a nightmare. And this storm, this is literally the same storm from NASA's website, there's West Africa. And it physically blew the locusts away. And this is how locusts actually get transmitted over very long distances. It's known, in fact, if you look at the genetics, that locusts in America did come originally from Africa. So it was a disaster. 2 months work, 20 minutes worth of data. I've been too traumatized ever to look at it. I still have the video, but I will never look at it. So I came out with my collaborators to the good old US of A, where you have things like roads. So we came to Utah and Idaho to study a similar type of organism, the Mormon cricket. So here you can see a Mormon cricket chowing down on one of his buddies, but it's dangerous to do that. So they'll tend to carry them along, try and find something to climb up, a bit like a jaguar taking prey up a tree to get away from competition. Why are they called Mormon crickets? Well, you may be interested to know this is the monument outside the Mormon temple in Salt Lake City. But the Mormon pioneers, just when they were settling in, things were going great, and then suddenly this huge plague of crickets came, and they were going out with dogs and fire and just trying to get rid of these crickets. And then God sent a huge flock of seagulls that ate up these crickets. And in fact, the seagulls do still come in from the Great Salt Lake to eat the cricket swarms. And so that's why they became known as Mormon crickets. So these apparently vegetarian insects, we'd see them eating roadkill. These jackrabbits, the ears are normally about this long. They'd eat the ears, they'd crawl in through the eyes and in through the mouth. Really gruesome. These are really hardcore organisms, and they'd eat each other as well. And so Steve Simpson had these artificial diets. This one's got protein and carbohydrate. This one's got carbohydrate. This is what you'd expect vegetarian insects to go for. This one's protein, and this one's got no nutritional value whatsoever. Well, they don't care about carbohydrate. Look, it's the same as the no nutritional value, but they love the protein. And we also did another experiment with salt, and this is my little living histogram. Of course, I randomized it for the experiments. But this is water, and this is increasing concentration of salt to 2 molar salt concentration. Look at them fighting over 0.25 molar. They really love it. They can taste it with their feet, and they fight each other over it. And that's the one that they love. And of course, when we measured the blood, that's exactly the concentration of cricket blood. So they're really fine-tuned to try to have a cannibalistic lifestyle. So we have this new mechanism of collective motion, the forced march, where individuals are attacking those ahead and trying to prevent themselves from being attacked from behind. Stop and you risk being cannibalized. So we can make predictions. If we give them protein and salt, we should reduce cannibalism and inhibit marching. And I won't go through these experiments, but with very strong significance, we were able to do so. But of course, finding the insect is the real problem. So understanding the driving mechanism now allows us to go to satellite data to try and work out where is protein lacking and where are swarms growing. Now, if you can find them, you may as well use pesticides, but this gives us good clues as to how we can understand it. And to come back to locusts, I don't have time to talk about this, but we've basically shown that this same theory holds for why locusts swarm. And it's kind of cool. Remember I described this idea from the magnets? This idea from physics of how magnets work, and we sort of stole that idea and applied it to biology. Well, one of the cool things about this chasing behavior of the locusts, we were then able to go back to the physics community and come up with a new mechanism for collective motion and for phase transitions in physics. So that was kind of fun. And the last thing I want to tell you about comes back to the evolution. No one really knows why you get these two morphs of locusts. In fact, the only explanation that's currently published explanation is a group selectionist argument. So we wanted to ask, well, can this cannibalism explain the evolution of these phases? And so what we do is we create a simulation model where individuals can evolve any behavior they want, any type of attraction or repulsion or whatever. And what we find is that below a critical density, they evolve to escape from everybody, i.e. avoid everybody. Above a critical density, they escape from those moving towards them, and they run towards those moving away from them. So they only know the relative optical flow. And so we have the transition between solitarius and gregarious, between no swarm and swarm. So we think we finally understand why we get these two forms of locusts. And I'd like to thank you all very much for listening. I would like to take questions, but I'm afraid I can't see too much up here. But if anyone has any questions, please yell them out. Mm-hmm. Well, I mean, one has to be kind of cautious. So we are doing experiments with people making consensus decisions. And my colleagues at Princeton in psychology are performing these experiments, and they have very positive results. But of course, if you start dealing with the real details of political scenarios and complex scenarios, then this type of modeling is not appropriate. But we certainly would like to try and understand at what level, say, small committees or juries, where this type of decision-making may actually be quite pertinent. Yeah, I mean, this is a very valid point. I mean, you can see it the other way, where a small subset actually is you want it to propagate. It's, they're being inhibited, but they actually have the right idea, and they're trying their best, but they're not able to do so because of this large number of uninformed individuals. So I'm aware of that, that you could see it from the other perspective, that the system could actually become into a stable state that is an undesirable state also. I don't think so, no. I mean, what we're really doing here is trying to understand the evolution of, actually, microevolution of migration occurs within decadal timescale. So it's actually remarkably fast examples of evolution. And so those are the types of scenarios that we're looking at. I think if you start dealing with humans, there are all sorts of other factors that we don't take into account within these types of models. And so I wouldn't want to suggest that this work goes beyond, sort of ungulates and migrating birds and so forth, or migrating cells. That's really what we're focusing this research on. I'm not suggesting that it's not interesting to look at human migration. It would be fascinating, but I would need to know a lot more.
You talked a lot about uninformed individuals and groups. Did you experiment at all with misinformed individuals?
Yeah.
Is there such a thing in the animal world, and if so, what were your results?
Absolutely. There is very much a thing of misinformed individuals, and I would say that that's something that we are working on now, but we don't have good concrete results. But there's a chap called Kevin Leyland at the University of St Andrews in Scotland who did experiments whereby he trained fish there are two routes to the food. One is long and one is short. And so he trained the fish to take the long route. And then he started exchanging the trained fish with fish that didn't know what was going on, but they copied the trained fish. And so you have a sort of culture, so to speak, of taking the long route, even though the short route was there. By giving them the wrong information to begin with, that can be stuck in the system until, this is where randomness makes an important point. If a fish makes a sort of mistake and accidentally finds the short route, then the system can quite quickly flip to use the more efficient route. So this is why in nature, we sometimes wonder, why do we have so much randomness? Well, randomness allows you to explore different solutions. But it's a really interesting question, and it's something that I'd like to look at more.
Do you have anything about how flocks will interact with other flocks when they don't want to merge? Like, how will they stay the right distance away to distribute themselves across an ecosystem?
Right. So most of the systems that we look at, say the schooling fish, are in what's called a fission-fusion dynamic, whereby the groups will very frequently merge together and split apart. And so there's lots of mixing going on. But even within that scenario, if you actually sample a fish school and you measure the individuals, they're very similar in length to each other. And they may be very different to another fish school. And so biologists used to think that they sort of know how big they are, and they actively aggregate with others that are the same size as them. But what we now know from our modeling is, in actual fact, you spontaneously get sorting. It's a bit like, you shake your muesli, and the big bits go to the top and the small bits go to the bottom. Within these biological examples, these animal groups are self-stirring fluids. If you put a drop of milk in your coffee, it takes about 2 days to diffuse. If you put a drop of milk in your coffee and you stir it, it's almost instantaneous. So these flocks are self-stirring media, which means they structure themselves spontaneously. And so that allows groups to sort of form structures. But we haven't studied any scenarios. I'm sure primates is a classic example of where the groups really have these competitive interactions, but I haven't studied that myself. So we're not tracking the motion of the eyes, unfortunately. The resolution isn't there. But the fish, fortunately for us, has a very strict geometry, as we do. The eyes are in a very predictable place. So we track the fish, we fit a spline to the body, which maps the curvature of the body, and then we use the geometrical relationship, which we've measured from many experiments, to pick out where the eyes are. So the question was, my understanding of the question was, what sort of implications does this have in the medical fields in terms of cell movements and cell tracking? Well, we think, for example, that the hypothesis, that tumors, and it's not only us that's coming up with these types of ideas, that tumors are sort of fast-evolving biosystems, within bodies, and there's strong selection pressure. And so when the tumors grow, there's, they effectively run out of resources. And once that happens, there's a very strong selection pressure to get out of that nutrient-poor environment. And so that seems to be the driving force for these cells that will actually start migrating. And in most tumors now, it's now known that this sort of classic view of a single cell going off into the bloodstream and forming a tumor is not what happens. They actually go off in rafts or in tendrils, and there's a strong collective component. There's also an interaction with the immune system that's important. So we're beginning to work on that with some colleagues at Sloan Kettering. But it's early days, but I'm dead excited about it.
What, if anything, can you infer about speciation rates of migratory versus non-migratory species?
That's an excellent question, and there's a recent paper that I reviewed that's just been published that looked not at speciation, but looked at birds' migratory passerines or songbirds and look sort of back through evolutionary time to sort of see where did sort of long-distance migrants come from, and where did species, that were long-distance migrants become short-distance migrants and so on. But that didn't explicitly look at speciation. I have colleagues at the University of Florida who argue they've modified our model, and they argue that this collective behavior allows individuals to find solutions and therefore speciate faster. But I haven't seen that work because it's not been published yet. But it's something that they're working on. It's a good question. I don't know where to go. Maybe this.
What have you learned from this work that contributes to the success of the whole? What contributes to the survival or the success of the whole?
Yeah. Well, the whole organism or the whole group?
The whole. Is there a difference?
There's a huge difference. Think of the locusts, right? The locusts, it looks like they're all coordinating and cooperating, but everyone is actually trying to eat everyone else. It's been a huge danger in biology to think about group level selection for the majority of types of groups that I've been talking about. That's not to say all groups. And certainly, there's been great progress made in understanding human societies by looking at intergroup conflicts and primates and so forth. But for the types of groups I'm looking at, where there's a high degree of fission and fusion and mixing, then you really have to think about the individual being the level of selection. But there's still some intriguing issues about how individuals, just by trying to optimize their own behavior, can actually facilitate group-level optimization. It's one of the biggest challenges at the moment, is trying to understand under what conditions do selfish individuals actually evolve to have effective, optimal group-level behaviors. Okay.
So when the locusts or the kids are in this cannibalistic phase, is that when they're actually swarming and out eating crops?
So the amazing thing is people have often reported that the swarms will move through crops without apparently, what look like perfectly good crops without eating them. And what we found in the Mormon crickets is that the leading edge of the swarm are eating crops, are eating plants, but strictly the proteinaceous parts, the flower heads, the pollen. Okay, so they're really limited in those resources. And so this could explain, this cannibalistic drive could explain why you're moving through the carbohydrate-rich areas. That's not what you need.
The fact that uninformed individuals enhance collective behavior on some factors theories, for example, in the case of financial markets.
It is deleterious, you said?
Or, yeah, because
What?
people have claimed that most of the financial cracks come from this herd behavior, right?
Right. But the uninformed individuals in this case aren't just blindly, aren't just acting in that type of fashion. I mean, I think one has to be cautious. Of course, one talks about herd dynamics within financial markets, but of course this model isn't a model of those types of herd dynamics. So I think one would be cautious about making a direct link there.
Okay, well, let's thank Ian for a great talk.
Thank you.